differentiate somatic embryogenesis and zygotic embryogenesis. See, first we discuss about the somatic embryogenesis. Somatic parts means vegetative parts of plant body. Vegetative parts of plant body may be leaf, shoot, root. These three parts are called vegetative parts. So the vegetative parts are also called somatic parts of the plant. From these three parts, from leaf, from shoot, from root, we take any cells, they are in diploid condition. The diploid condition cells are having totipotency character. Each somatic cell is also called a sporophytic cell of plant. They can ability to development of complete plant by single diploid cell. So we collect the explants from root, shoot, leaf. That explants are transferred into the nutrient medium. That explants generate or produce callus. Callus is a mass of cells present on the nutrient medium. If we observe callus on the nutrient medium, if we add growth hormones like auxins and cytokinins to the callus, they are convert into the callus cells are convert into globular structure, heart or torpedo structure of embryo, and that structure develop into organogenesis. Mean complete plantlet, whole plantlet is regenerated from the vegetative cells. So this process is also called somatic embryogenesis. We selected single cell or mass of cells from the leaf, root, shoot of the plant. The tissues are called somatic tissues, transfer into the nutrient medium, develop into callus. Callus identified on the nutrient medium, we add growth hormones like auxins and cytokinins. It develop into organogenesis. Before the organogenesis, they develop into formation of embryo from callus. So bipolar development, bipolar generation. From this heart or torpedo embryo, root is developed, shoot is also developed, complete whole plant is developed. So regeneration of whole plant from vegetative parts called somatic embryogenesis. It completely under aseptic conditions maintain proper environment to the somatic embryogenesis procedure. But we take zygotic embryogenesis. Zygotic means zygote, the fertilization, true fertilization or syngamy, fusion of male gamete and egg cell to form zygote is called syngamy. By the syngamy or by the fertilization, diploid zygote is formed in the ovule. After fertilization, the ovule convert into seed. Near the zygote, primary endosperm nucleus is present, and outside of these embryo sac, perisperm tissues are present, and perisperm tissues are also called nucellus tissues before the fertilization. But the nucellus tissues are called after fertilization perisperm, and primary endosperm nucleus, it formed by the fusion of male gametes and secondary nucleus. Finally, primary endosperm nucleus develop into endosperm, perisperm. These two types of tissues are provided nutrients to zygote. So zygote naturally received nutrients from the endosperm or from the perisperm. The zygote multiplies into cells, develop into globular, heart-shaped or torpedo-shaped, and finally dicotyledons or monocotyledon embryo is developed from the single zygote. These embryos are protected by seed coat. Seed coat is protected to embryos. But in somatic embryogenesis, seed coat is absent. But in zygotic embryogenesis, seed coat is present. Below the seed coat, embryos are present. The seed codes containing embryos means completely matured seeds, expansion, maturation, desiccation, and dormancy. Desiccation after dormancy. So after dormancy, the seed develop into germination, germination of plant, and it is involves in the growth. So at development of embryogenesis, zygotic embryo is involved in the expansion, maturation, desiccation, dormancy. After these complete procedures, after the dormancy, the zygotic embryo develop into complete plant, all also called germination of plant. And by this differentiation between zygotic and somatic embryogenesis. See, zygotic embryogenesis formed by the fertilized egg. Fertilized egg is also called a zygote. But in somatic embryogenesis, we take diploid sporophytic cells from the vegetative parts of plant like root, shoot, leaf. And a zygotic embryo contains seed coat. Somatic embryo did not contain seed coat. Zygotic embryo produced seeds. In somatic embryos only from to form embryos only embryos, not seeds. In zygotic embryo, plantlets are in healthy condition. In somatic embryos, plantlets are weaker because the somatic embryogenesis is completely under aseptic condition. Always we provided proper environment, proper temperature, proper gases, and including nutrient medium to the plantlets. So if we are not provided these such types of things, the plantlets are shows weaker. But in zygotic embryogenesis, they are having sufficient nutrients around the plants. And zygotic embryos, not like two mother cells, not like two mother plant. And the somatic embryos like to be mother plant because the somatic cells collected from the vegetative parts of plant body. So somatic embryos develop into identical parental plant. But in zygotic embryos, not identical to the parental plant or mother plant. And the zygotic embryo propagation is very low, and the somatic embryo propagation very high because the propagation means multiplying of plants on the nutrient medium, development of identical plants from the callus. So it is high propagation of somatic embryogenesis. These are the main themes of the zygotic embryogenesis and the somatic embryogenesis and their differentiation.